Good day, doctor. My name is TENG CHUN. Today I will be presenting the topic entitled Gut Microbiome and Human Health. This presentation will introduce what the gut microbiome is, its importance in maintaining human health, how dysbiosis contributes to disease, and how current research and therapies are advancing this field. Let's begin with the definition of the gut microbiome. The human body is home to trillions of microorganisms, including bacteria, fungi, viruses, and archaea. These microorganisms form a mutualistic relationship with us, meaning that both the microbes and also the human host benefit from each other. The gut microbiome refers to the collection of microorganisms living in our gastrointestinal tract together with their genetic material and also their metabolic products. Unlike our organs, the gut microbiome is not fixed. Instead, it is a dynamic ecosystem that continuously changes throughout our lifetime and is influenced by factors such as diet, age, lifestyle, medications, and also the environment. Among all the microbial communities in our body, the gastrointestinal tract contains the highest density of microorganisms. In healthy individuals, these microorganisms exist in a balanced and stable relationship with the host. A healthy gut microbiome is characterized by high microbial diversity, rich genetic content, and a stable core community. This balance allows the gut microbiome to perform many essential biological functions while preventing harmful microorganisms from dominating the intestinal environment. The gut microbiome has three major functions. First is nutritional metabolism. Gut bacteria help to digest dietary fiber that humans cannot digest on their own. During this process, they produce beneficial metabolites such as short-chain fatty acid, which serve as an energy source for intestinal cells. Second is immune system development and also homeostasis. The gut microbiome trains and regulates our immune system, helping it distinguish between harmless microbes and also harmful pathogens while preventing excessive inflammation. Third is neuroendocrine regulation. Through the gut-brain axis, gut microorganisms communicate with the brain by producing neurotransmitters and also signaling molecules that will influence mood, behavior, and also brain function. When the balance of the gut microbiome is disrupted, this condition is called gut dysbiosis. Dysbiosis has been increasingly associated with many human diseases. Studies have shown links between altered gut microbiota and inflammatory bowel disease, metabolic disorders such as obesity and also diabetes, neurological disorders, and even cancer. This demonstrates that maintaining a healthy gut microbiome is important not only for digestion but also for overall human health. Some examples include inflammatory bowel disease, especially Crohn's disease, and also ulcerative colitis. Gut microbiota imbalance is also associated with metabolic diseases, including obesity, type 2 diabetes mellitus, non-alcoholic fatty liver disease, and also cardiovascular diseases. Furthermore, increasing evidence suggests that the gut microbiome disturbance may influence neurological disorders such as Alzheimer's disease, Parkinson's disease, autism spectrum disorder, multiple sclerosis, and even stroke through the gut-brain axis. To better understand dysbiosis, let's focus on ulcerative colitis. Ulcerative colitis is a chronic inflammatory bowel disease that mainly affects the rectum and also the colon. It causes persistent inflammation of the intestinal lining, resulting in symptoms such as abdominal pain, diarrhea, rectal bleeding, and also weight loss. Although the exact cause is still unknown, it is believed to involve interactions between genetics, environmental factors, immune dysregulation, and also gut microbiota imbalance. Patients with ulcerative colitis show approximately a 25% reduction in gut microbiome diversity compared with healthy individuals. Specifically, beneficial bacterial groups such as Firmicutes and Bacteroidetes are reduced, while potentially harmful bacteria such as Proteobacteria and Actinobacteria become more abundant. Importantly, beneficial butyrate-producing bacteria, including Roseburia hominis and Faecalibacterium prausnitzii, are significantly decreased. Since these bacteria produce butyrate, their reduction contributes to disease progression. Short-chain fatty acids, especially butyrate, acetate, and propionate, are important metabolites produced by gut bacteria during fiber fermentation. These molecules maintain intestinal barrier integrity, provide energy for colon cells, and also regulate immune responses. In ulcerative colitis, FCFA levels are significantly reduced. Lower FCFA concentrations weaken the intestinal barrier, impair immune regulation, and promote chronic intestinal inflammation. Interestingly, patients in remission usually have higher butyrate levels than patients with active disease, highlighting the protective role of FCFA. After the gut microbiota becomes disrupted, the balance between beneficial and harmful bacteria is lost. As a result, the intestinal barrier and its immunoregulatory capacity become impaired. This weakened barrier reduces the host defense against pathogens, allowing harmful bacteria to rapidly proliferate in the intestine. These bacteria can then invade and also damage the intestinal epithelial cells. Once the epithelial barrier is compromised, microbial antigens and bacterial products are able to penetrate the intestinal tissue. This increased infiltration of microbial antigens sets the stage for an aggressive immune response, which will be explained in the next slide. When the microbial antigens penetrate the damaged intestinal barrier, they are recognized by toll-like receptor located on the intestinal epithelial cells and also the immune cells. Activation of these receptors trigger the MyD88 signaling pathway, which subsequently activates the NF-kappa B signaling cascade. NF-kappa B functions as a transcription factor that will stimulate the production of pro-inflammatory cytokines, including TNF-alpha, interleukin-6, and also interleukin-1 beta. These cytokines amplify the inflammatory response, leading to extensive intestinal inflammation. As inflammation progresses, even more microbial antigens infiltrate the tissue, creating a vicious cycle that exacerbates tissue damage and contributes to the progression of ulcerative colitis. One promising treatment for gut dysbiosis is fecal microbiota transplantation, which is also known as FMT. FMT involves transferring processed stool from a healthy donor into the patient's gastrointestinal tract. The goal is to restore a healthy and also diverse microbial community in the patient's gastrointestinal tract. FMT can work through several mechanisms. First, it restores microbial diversity and allows beneficial bacteria in the gastrointestinal tract to compete with the harmful microorganisms for nutrients and also intestinal niches. This process is called as competitive exclusion. Second, healthy microorganisms stimulate repair of the intestinal epithelium, increases mucus production, promote antimicrobial peptide secretion, and also restore immune balance. A successful real-life application of FMT is in recurrent Clostridium difficile infection. Following antibiotic treatment, the normal gut microbiota is disrupted. This will then allow the Clostridium difficile to overgrow in the patient's gastrointestinal tract. FMT will restore colonization resistance and also effectively suppresses the pathogen, which will then reduce the recurrence rates in the patient. Besides treatment, researchers also need tools to understand the functional activities of the gut microbiome. One powerful technique is metaproteomics. Unlike sequencing methods that identify which microorganisms are present, metaproteomics identifies the protein that microorganisms are actively producing. Therefore, it provides direct information about microbial function. The process of metaproteomics begins with protein extraction from stool or intestinal samples, and these proteins will then be digested into peptides and separated by liquid chromatography, analyzed using data mass spectrometry, and then matched to the protein databases for identification. A recent study by TENG CHUN and colleagues analyzed fecal samples from 134 healthy individuals by using liquid chromatography tandem mass spectrometry. The researchers identified 182 core microbial function and 83 metabolic pathways, including pathways involved in glucose metabolism and butyrate production. This study demonstrates how metaproteomics help us better understand healthy gut function and also identify potential biomarkers for the disease. Here is the reference that I used to gather all the information about the gut microbiome and also the ulcerative colitis related to the disturbance of the gut microbiome. In conclusion, the gut microbiome plays an essential role in maintaining human health through nutritional metabolism, immune regulation, and also neuroendocrine communication. Destruction of this microbial ecosystem, known as dysbiosis, will contribute to numerous diseases, particularly inflammatory bowel disease such as ulcerative colitis. Therapies such as fecal microbiota transplantation offer promising approaches to restoring microbial balance, while advanced techniques like metaproteomics enable researchers to better understand microbial function and also develop future precision medicine strategies. Thank you very much for your attention. That's all of my presentation.